Hello and welcome to The Explainer. So today we're going to play detective. We've got this tiny tube of clear liquid, and it's supposed to have our precious DNA in it. Our mission, if you choose to accept it, is to figure out what's really inside. How much is there, and is it clean enough to actually use? I mean, look at this thing. You go through all the trouble of a DNA extraction, and this is what you're left with. This little tube could hold the key to a groundbreaking experiment, a perfect sequencing run, maybe a successful cloning project, or it could be packed with invisible enemies, contaminants that are just waiting to ruin everything. So how do we find out the truth?
Okay, first things first, let's talk about the stakes here. Quantifying your DNA isn't just some optional step you can skip. Honestly, it's one of the most critical quality control checks you can do. If you get this part wrong, you could waste so much time, so many expensive reagents, and you could end up with results that mean absolutely nothing.
Just think about it for a second. If you're running a PCR, you need to add just the right amount of template DNA. For a restriction digest, all your enzyme calculations are based on how many micrograms of DNA you have. And for something super sensitive like next-gen sequencing, you have to know exactly what you're putting in. The success of all these techniques really hinges on knowing you've got a specific amount of pure DNA to start with. So how in the world do we measure something that's completely invisible? Well, it turns out DNA has a secret signature, a tell. We just need to know how to look for it. And the clue is all about how it interacts with light. This is the science that's going to power our entire investigation. Now, the principle behind this is called the Beer-Lambert law, but don't worry, we can skip the complicated math. The simple version is this. Imagine shining a flashlight through a glass of water. Now, if you start dissolving something in that water, the light on the other side gets dimmer, right? The more stuff you add, the more light gets blocked. That's it. Our tool, a spectrophotometer, is basically just a really precise light meter that measures how much light gets absorbed. Okay, but what kind of light are we talking about? Well, it turns out that nucleic acids, that's both DNA and RNA, have a favorite color of light that they absolutely love to soak up. It's not a color we can see, it's in the ultraviolet spectrum, specifically at a wavelength of 260 nanometers. This is our key clue. It's the unique fingerprint we're searching for. But here's the thing. Our target, the DNA, rarely travels alone. It usually has some accomplices, contaminants that are left over from the extraction process. But luckily for us, these accomplices have their own light fingerprints too. Proteins, a really common one, absorb light most strongly at 280 nanometers. And other chemicals like phenol or salts, they show up at 230. So by checking these other wavelengths, we can build a pretty good profile of who else is lurking in our sample. Now, the go-to tool for this whole operation is the nanodrop spectrophotometer. You should really think of it as our high-tech magnifying glass. And the reason it's so popular is because it's super fast, incredibly easy to use, and get this, it only needs a tiny drop of your precious sample. And when I say it's easy, I really mean it. First, you just wipe the little sensor pedestals clean. Then you add 1, maybe 2 microliters of your blank, which is just the buffer your DNA is dissolved in, to zero out the machine. You wipe it again, add 1 single microliter of your DNA sample, close the little arm, click measure. And that's literally it. Within seconds, you've got your results. So the nanodrop spits out a concentration number, which is great. But that's not the whole story. Not even close. The real detective work, the part where we crack the case, is in decoding 2 crucial ratios that it gives us. These numbers tell us all about our sample's purity. All right. The first and most famous of these is the A260 over A280 ratio. This is our #1 detector for protein contamination. For a really pure DNA sample, you're looking for a number in the sweet spot, right between 1.8 and 2.0. If that number starts to dip below 1.8, that's a big red flag for protein. And if it's way over 2.0, well, you might have an RNA problem, since RNA also absorbs light at 260. And check this out. Here's a real-world example from one of our sources. The machine read out a 260-280 ratio of 1.53. That number immediately tells us, whoa, hold on, this case is not closed. A number that low means the sample is significantly contaminated, probably with protein, and you've got to clean it up again before you can trust it in any important experiment. Okay, so we've checked for protein, but our sample might not be a solo act. There could be other accomplices hanging around. To find them, we've got to look at our second clue, which is a whole different purity ratio. And that is the A260 over A230 ratio. This one is our detector for all the other junk, things like salts or phenols or carbohydrates that might have carried over from the extraction kit. For this ratio, what you really want to see is a value of 2.0 or even higher. If that number is low, it means you have contaminants that can mess with your enzymes later on. Okay, so let's imagine you nailed it. Your concentration reading is fantastic. Your 260/280 ratio is a beautiful 1.85. Your 260/230 is 2.1. Everything looks perfect, right? You're ready to go. Well, not so fast. There's a plot twist. And this is where our whole investigation takes a sharp turn. Because as amazing as this tool is, it has one massive blind spot. It can be fooled. There is one critical piece of information that the nanodrope absolutely cannot give you. And this slide lays it out perfectly. The nanodrop is fantastic at telling you about quantity, how much stuff is in there, and purity, if that stuff is contaminated. But what it is completely blind to is the DNA's integrity. It has no way of telling you if your DNA is made of long, beautiful, usable strands, or if it's just a bunch of worthless, tiny, degraded fragments. Let me say that again because this is probably the most important takeaway. To the spectrophotometer, a single free nucleotide, a short, broken piece of DNA, and a giant, intact chromosome all look exactly the same. They all absorb light at 260 nanometers. The machine just adds it all up and gives you a total concentration, which can give you a very dangerous, false sense of security. So, while we may have solved the mystery of quantity and purity, one final, crucial question is left unanswered. Your sample might be pure, but is it intact? To answer that, you need another tool, something like gel electrophoresis, to actually see the quality. So remember, the case is never truly closed until you've confirmed your DNA isn't just pure, it's also whole.